Hi, and today we're going to start looking at some of the more tricky stuff within the kidney. We're going to look at ultrafiltration and selective reabsorption. So first of all, here's just a little couple of exam questions for you. So if you just want to have a pause again and see if you can work out what these are, there are also past paper questions in your past paper question booklet. So you may have done these ones, you should have done these ones already. So if you just want to have a pause, and then we'll go through them. Right, so A, it says name the type of blood vessel labelled B, and in the blurb at the top it says it is a photomicrograph through the centre of a lobule in the liver. So if it is in the centre of a lobule, then that means that is the central vein. And the question says the type of vessel, so the type of vessel should just be vein. If you put hepatic vein, you do not get the mark because that is not the hepatic vein. If you put central vein, you still get the mark, but actually it should just be vein. And then the type of cells that make up the lobule, you can either put hepatocytes or you can put liver cells. You do get the mark for liver cells, but obviously you should at this stage be putting hepatocytes. So there's another question on the next slide. So again, if you want to pause, have a little think about this, and then we'll go through it. Okay, so C, what is process C? It is the formation of the ammonia from the amino acid, so that is deamination. Compound D, now you can see we're going from ornithine to citrulline. We've already got ammonia coming in here. So compound D cannot be ammonia because we've already got that. We've got the water coming out over here, so compound D must be the carbon dioxide that's eventually going to form the urea. Now I'm going to not go in order because I'm going to come round to F because it kind of makes sense in the ornithine cycle. Now the first thing to notice is that this ornithine cycle is going that way around, it's going anti-clockwise, whereas in your diagram, in your book, it was going clockwise. So please, please, please be aware of that. So from citrulline to arginine, you can see again we already have an ammonia coming in here. So compound F obviously can't be ammonia, it's coming out anyway. So we've got ammonia going in, our second ammonia. So from citrulline to arginine, again, it is a condensation reaction. So compound F must be water because it's a condensation reaction. So then from arginine to ornithine, you've got the water here going in. Now you know the whole point of the ornithine cycle is to the formation of urea. So compound E must be urea. And if you looked at the formula for that, it is CONH2 twice. So you can see that there is your carbon dioxide, there is one ammonia, and then the second one going in there. So you've got two ammonias and you've got your carbon dioxide there. Okay, so hopefully you got those right. And like I say, it's in your past paper question booklet as well. Okay. Right, so in this particular presentation, we're going to have a look at ultrafiltration and we're going to have a look at selective reabsorption. So first of all, let's have a look at the language. So ultrafiltration, you know what filtration is? Filtration is taking things out of something else. So taking the solutes out of a liquid. This is ultrafiltration. So as the name suggests, we're gonna filter out a lot of stuff. We're going to filter out pretty much anything that we can. And then the selective reabsorption. So reabsorption is taking things back. Selective, we're going to take certain specific solutes, ions and water back. So this is how the good things, the things that we want to keep, are taken back. So we're going to look at these two systems separately, these two processes separately. So you can have a go at doing that if you want to. You can always check back with further back in your book to see if you've got that right. You should be able to draw one. You should be able to draw an nephron. You should be able to label all of those things. You should be able to label all of it. Right, so again, if you want to check, if you want to have a look, see if you've actually managed to do that correctly, then you can have a look. So, ultrafiltration, selective reabsorption, two main functions of the kidney. Well, you've got getting rid of the urea. We've already talked about that one in the formation of the urea in the liver. We talked about it then being got rid of in the actual kidney. But the other main function of the kidney is actually water control. So something called osmoregulation. We're going to come to lesson after next, I think, or maybe next lesson. Anyway, osmoregulation. Shall I spell that out? So it is osmo, because we know that's to do with water, osmosis, osmotic pressure, osmoregulation. We're going to control the water. Now, as you've seen before, and as you know from looking at water potential and water potential gradients, you can't move water unless you also move ions. So water balance and water control, you can see there that it's written as water/mineral balance, because water and ions have to be controlled together. Okay, so they're the two main functions of the kidney. So ultrafiltration and selective reabsorption is a way of getting rid of the urea. Now, you can't get rid of all of the urea, and you'll see that later, but you can tolerate a small kind of concentration of urea in your blood. You just don't want too much. So you just need to get rid of most of it. You don't have to get rid of all of it. So ultrafiltration. Your body does not have a way of picking out urea. Urea is soluble, so it is a solute within the plasma of the blood, and so as it's going through the kidney, I mean, as we saw the, did we look at the flow rate? We're going to look at the flow rate in a different lesson, actually. The flow rate of blood through the kidney is quite high, and so just trying to pick out the urea, we don't have a way of doing that. And we don't need to pick out urea anywhere else in the body, so it would be a mechanism that would be specific to the kidney. So it would have had to have evolved very, very separately. And as we know through evolution, it's a bit of a messy process. We evolve things that do the job not necessarily in the best way, but that's how it evolved, and it worked. So why bother fixing it? If it ain't broke, don't fix it. Good is good enough, as we say to teachers. Right, so in ultrafiltration, nearly everything is pushed out. So it's a little bit like the tissue fluid formation. We are not going to be pushing out large proteins. We are not going to be pushing out red blood cells or white blood cells, but we are going to be pushing out glucose, amino acids, and much smaller proteins, and you'll see in a minute which ones. We are going to be pushing out ions, and we are going to be pushing out pretty much anything that's small enough. So that's why it's called ultrafiltration. Now, once you've pushed this stuff out of the blood and it is then in Bowman's capsule, it is then known as filtrate. So we're going to look at the formation of filtrate in ultrafiltration. So once you've pushed all of those things out, now obviously some of those things you want to keep. You are going to want to take back the glucose, you are going to want to take back the amino acid, you're going to want to take back the majority of the water, otherwise you would very quickly dehydrate and die. But we already have ways of moving those. We have ways of moving glucose and amino acids, we have ways of moving water, because we have ways of moving ions. So we have the ability to selectively pick these things back out of the filtrate. But what doesn't get reabsorbed is the urea, and that will stay in the filtrate, and then you'll kind of eventually get rid of that through the collecting ducts, through the ureter into the bladder, and that will then be urine. So let's have a closer look at the glomerulus. Now, I like this diagram because if I can actually point very carefully, you can see that inner grey layer there. Didn't quite get it there. Yeah. That is the inner layer of Bowman's capsule, and that's what I was saying about there not being a gap between the blood vessels, that knot of capillaries. So you can see here, this is the glomerulus, this network of capillaries in here. There is no gap between that and Bowman's capsule. So Bowman's capsule and the glomerulus are kind of kind of, really tightly associated with each other. There is a space, however. So here is a space between the inner part of the glomerular of Bowman's capsule and the outer part of Bowman's capsule. The Bowman's capsule, God, what could you think of it as? I have no idea. I can't think of an analogy that would really work for that. Well, anyway, so you've got this inner layer and this outer layer, and then, as I said, there is a space between them. So you've got the inner layer, you've got the outer layer, and then you've got this space here. So anything that gets pushed out of the blood gets pushed out of the blood into this space and will eventually drain down here. So it gets pushed out of the blood, 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 forms the filtrate in this space, which will eventually drain down into the proximal convoluted tubule. Okay, now this picture over here is actually lovely. So what happened here is that they filled all of the blood vessels and then digested away the tissues. So these, if I can highlight these very tight ones here, try to pick a different colour. These very tight ones, they are the glomerulus. These slightly looser ones, these are the capillaries that wrap around the proximal convoluted tubule and the loop of Henle. So they're not quite such a knot, but that just shows you how much of a blood supply you get in the kidney. So you can see you've got this tight association. Now this inner layer that I talked about, this inner part of Bowman's capsule, has a layer of cells called podocytes, which are actually wrapped right the way around the capillaries. So we're going to have a look at those as well. So you have a diagram on page 24. You actually have this diagram on page 24. So if you want to, you can label that a little bit more. Don't worry about all the different arrows and what they are. You don't need to worry about those. You just need to worry about the blue ones, which is showing you ultrafiltration. Okay. So before we move on, I forgot to point out the afferent and efferent arteriole, and you can see very nicely on here that they do have that different lumen size. So your afferent arteriole has a much wider lumen than your efferent arteriole, and that is so that you have a lot of blood going in and the blood can't get out as quickly, and that generates pressure, and that pressure is what pushes these solutes and the fluid, the water, out of the capillary and into that glomerular space. Okay. Right, so you can see on this one, they are the podocytes and they are wrapped around. So these podocytes are the inner part of Bowman's capsule. They're not part of the capillary, they are part of Bowman's capsule. Okay, now the capillary is just like any other capillary. It's made of endothelium. It is one cell thick, very thin, squashed out epithelium, and they would burst. With that much pressure, those capillaries would burst. So those podocytes are tightly wrapped around that capillary. There's enough gaps between them that the solutes and the water can leave. They're not watertight, but they are tightly wrapped around enough to prevent or help to prevent the capillaries from bursting. Okay. Now, one thing you do need to be able to identify is a glomerulus and Bowman's capsule from a microscope picture. And that's what this bottom one is here. Now they are very distinctive. They're distinctive because you can see that gap. You can see that space going all the way around it. And if the picture, tells you that it's an image from a kidney, then you shouldn't ever get that wrong. That space should tell you that it's Bowman's capsule and a glomerulus. We're going to have a look in a minute on how to decide if it is a proximal convoluted tubule or a distal convoluted tubule as well. So ultrafiltration. The blood will enter that glomerulus through that afferent arteriole, so that's obviously branched off the renal artery. And like I said, it's under high pressure because of the differences in the diameter of the lumen. So the efferent arteriole is narrower. Now that filtration pressure forces out anything that is small enough. Now the cutoff point is called the renal threshold, and the renal threshold for your kidneys is 69,000. So anything that has a molecular mass of less than 69,000 will be pushed out of the blood. Now it is important for you to remember that number. Quite often you get questions where it will tell you the size of a protein and it will say that this particular protein has shown up in the blood. So you can do a test for your urine and to look for proteins, and if you find protein in your urine, then that shouldn't be there, because those proteins are generally bigger than 69,000, so you shouldn't have them in there. So that would suggest that something has gone wrong with your ultrafiltration and that things are getting through that shouldn't. Now, it's not to do with the reabsorption, because they're big proteins, so that's to do with the ultrafiltration. So water, ions, urea, amino acids, glucose, vitamins, minerals, all of those things, they're all filtered out of that capillary into that space of Bowman's capsule, and so we now call that fluid with all of those solutes filtrate. So you have space in your book. All of this information on this slide goes next to that diagram that says figure 1 on page 25. Now you can see these 3 layers, so you can see this stuff here. Let's pick a different colour. This stuff here, they are those podocytes. That is part of the glomerulus. That's the inner layer of the glomerulus. You can see here, that's the outer portion of the glomerulus, and you can see there is the space between those two. So it is a very tight, there's nowhere else for this fluid to go, it can't escape. This diagram down here shows you the 3 layers. So that one, that red one there, that is the endothelium of the capillary. So that's the actual capillary. It has pores to allow some of these things through, just like every capillary. Every capillary in your body is the same. Now in the glomerulus, they form these tight knots of capillaries. Elsewhere in your body, you get networks, beds of capillaries. So there is a slight difference in the way that they are kind of structured, if you like, in how they're arranged, I guess. But in every other respect, they're the same. So you have these pores, fenestrations. Fenestra is French for window. So these little pores, these little holes, allow these things through. And then you've got this basement membrane here. Now that is part of the glomerulus, and again it has kind of slits in it, and it allows certain substances through, but it doesn't allow others. And then you've got the podocytes, and these podocytes have these kind of interlocking fingers. Now the role of the kind of the capillary endothelium and the basement membrane is to stop some things leaving and allowing obviously those smaller things to leave. The podocytes do that as well, but their main job, the podocytes' primary function, is to prevent bursting of the capillary. They are there to resist the pressure, and you can see on this diagram those interlocking fingers. So this would be one podocyte here, and this would be the interlocking fingers of a different podocyte. So yes, so you've got this information on the left hand side here that you need to write down in your book. So the endothelium, that's going to allow things through because it's got those pores. The basement membrane is going to allow certain things through, not others, and it's made of glycoprotein. And then you've got these podocytes, and they support the capillaries against that pressure. They help to resist that pressure. So if you want to just pause and get that information down in your book. Right, now, as it says, you don't need to know this, but it does show these podocytes very nicely, kind of here and here and here, those fingers that wrap around the capillaries. Okay. Right, selective reabsorption. Now, this is going to involve something called co-transport. We've come across co-transport before. When we looked at plant transport, we talked about co-transport in translocation. So translocation was where we moved the sugar into the sieve tube elements. And we moved hydrogen ions first, and then the hydrogen ions plus the glucose were allowed to move. It's going to be a similar situation here, but we're going to move sodium ions and then we can move glucose or amino acids. So you have come across this idea before. So co-transport proteins will only move a kind of a combination of things. It has to be the right chemical environment, and the ions help create that chemical environment. So selective reabsorption, it's to get rid of the urea, and it allows you to take the other things back. So selective reabsorption is the reabsorption of those important or needed solutes, but also the water. If you move the solutes, water will follow. Remember, you create a water potential gradient in the proximal convoluted tubule. So urea is not reabsorbed. Some, a little bit, will diffuse back into your blood because you've got such a high concentration in the filtrate. Some of it will, but not enough to kind of do you any harm. So you're only taking back the good stuff. Glucose, amino acids, hormones, vitamins, all that kind of stuff. Now, when you talk about reabsorption, remember you've got your proximal convoluted tubule that kind of wiggles. I'll just draw it as a wiggle, and you would have your Bowman's capsule there. Anything that is reabsorbed from the proximal convoluted tubule goes into that network of capillaries. If I draw the network of capillaries in a different colour, it kind of wraps all the way around. And so anything that is reabsorbed, we mean it is reabsorbed into the blood. So reabsorption means it leaves the kidney tubule. And remember, anything that's in the tubule, anything that's in that proximal convoluted tubule will eventually leave the body as urine. It will eventually leave. So if you reabsorb, you're taking it out of what will become the urine, out of the filtrate, back into the blood, and it will then circulate around the rest of your body and you get to keep it. So the proximal convoluted tubule, the cells lining the proximal convoluted tubule have microvilli, and that is to increase the surface area. So if you look at a proximal convoluted tubule, and if you looked at cells on the inside there, they would have microvilli. And obviously cells don't have gaps between them, but this is very difficult to draw. So, and they're terrible microvilli. But you get the idea, and this is filtrate, and this would be wiggling its way down there. So you've got that massive surface area for reabsorbing that filtrate back out of the tubule and into your blood. And obviously that's going to require active transport, and so you need mitochondria. Now if you move the solutes, then the water will follow. So water will be reabsorbed by osmosis. Okay. Now, the volume of water that is reabsorbed in the proximal convoluted tubule, it's about 60 to 65% of the water that you filter out is reabsorbed in that first part, in that proximal convoluted tubule. Now, that doesn't ever change. It doesn't matter how hot it is, it doesn't matter how much exercise you've done, it doesn't matter how much water you've drunk. That volume of water is always the same. The control of water, the osmoregulation bit, is in the collecting duct, and we'll do that in a different lesson. So the collecting duct is where you can reabsorb more water if, for example, it is a hot day or you haven't drunk enough, you are dehydrated, then you can reabsorb more in the collecting duct. So the control of water is in the collecting duct, but the majority of the water reabsorbed is in the proximal convoluted tubule. Okay. Right, so I did tell you needed to know the difference between a proximal convoluted tubule and a distal convoluted tubule. Now the proximal convoluted tubules, as I said, have microvilli. The distal convoluted tubules don't. So when you look at them down a microscope, the proximal convoluted tubules, let's see if I can, you can see one here, they look furry on the inside, whereas if you look at this one, which is a distal convoluted tubule, it doesn't look furry. The inside of this looks quite kind of clean you can see kind of individual cells, whereas this looks all kind of fuzzy and furry, and there's another one that looks fuzzy and furry, and there's another one that looks kind of fuzzy and furry, whereas the distal convoluted tubules, you can see that they're all these blue ones, they don't. They look really kind of nice and clean. And you can see over here as well, there's a proximal convoluted tubule. It looks kind of fuzzy, whereas the distal convoluted tubules, they look a little cleaner, a little crisper on their edge. Okay, and then this one shows very nicely a glomerulus with that Bowman's space of the Bowman's capsule. Okay, so take some time to have a look at this and make sure you can actually see the differences, because there have been exam questions where you've been asked to identify the proximal convoluted tubule or a distal convoluted tubule. I think you actually had to identify proximal, but have a look, make sure you actually know it. Okay. So how do we actually reabsorb? So you've got this diagram in your book on page 26, and you've got some stuff underneath it. So we're going to have a look at this process. So the first thing that happens is sodium is actively transported out of the cells of the proximal convoluted tubule. So let's just orientate ourselves in this diagram. So this kind of here, in here, that's inside the proximal convoluted tubule. So this is the proximal convoluted tubule. These are the cells that make that proximal convoluted tubule. Out here is a bit of space, just a bit of tissue in the kidney, and then in here you've got a capillary that runs, these are the capillary networks that run around these tubules. So anything that is reabsorbed from here and into the blood. Okay, so let's just get rid of all of these so you don't get confused. Right, so the first thing that happens is sodium ions are actively transported out of the cells that make up the proximal convoluted tubule, and you don't need to worry about the potassium, don't worry about that at all. So the sodium is actively transported out and eventually makes its way into the red bloods, into the capillaries. Now what that does is it lowers the concentration of sodium ions inside these cells. So what happens is sodium is drawn into the cells of the proximal convoluted tubules from the filtrate. So that filtrate inside the proximal convoluted tubule. So those sodium ions, when they move, they actually move through co-transport proteins, which is that represented by that thing. And when the sodium ions move through, they bring glucose with them. So this is a co-transport protein. So you move this sodium out, the sodium concentration inside these cells drops, sodium ions will want to move in down their concentration gradient, bringing glucose with them. Now there are co-transport proteins for glucose, there are co-transport proteins for amino acids. So the same process happens with amino acids. Yep. So then you move your sodium via your co-transport. Okay, does that make sense? Now once they build up in concentration, they will move by facilitated diffusion into your capillary. So glucose moves in, the concentration of glucose inside these cells rises, and so glucose will move by facilitated diffusion. That's what that protein is supposed to signify. Now, if I just get rid of all of this confusion for a second, you can see there, that is an active transport protein. It kind of looks like a little sun or a starburst, I guess. This one is different. So this one is showing you that this is just facilitated diffusion. Okay, so once you build up the concentration of glucose inside these cells, facilitated diffusion will then take them out of those cells and into the capillary. Now, what that does, because you've moved the sodium, you've moved the glucose, you've moved amino acids, you've moved solutes out of this fluid. And so what happens is the water potential gradient increases. And so water will move by osmosis out of the proximal convoluted tubule. Okay, you've created a potential gradient because you've removed those solutes, so you've increased the water potential inside the proximal convoluted tubule. Okay, right, so we have that in words on the next slide, and you can take that down in your book as well. So let's go through it. Filtrate enters the from Bowman's capsule. Sodium ions are actively transported out of the proximal convoluted tubule. You've got that pump. They're going to then diffuse into the capillary. It's going to lower the concentration of ions. So sodium ions are going to move in from the filtrate into the proximal convoluted tubule cells. They move through that co-transport protein, bringing glucose or amino acids, there's one for each, with them. Glucose is increasing in concentration. It's going to diffuse into the blood by facilitated diffusion. The water will move because of that movement of ions by osmosis. And by the end of the proximal convoluted tubule, all of the glucose and amino acids, about 65% of the water, has been reabsorbed. It's been taken out of that filtrate. It's not going to leave the body as urine. It's been taken back into the blood. So if you want to have another look at that, make sure you understand. So what's happening at 1, what's happening at 2, what's happening at 3, and as a result of that, what happens to the water. So just have a pause and have a little think about that, and make sure you can match the wording to the diagram. Okay, so this is a little quiz. If you want to pause, you can have a go at answering these questions, and then I'll go through them in just a minute. Okay, so ultrafiltration, it is pushing out the majority of the stuff, really, pushing out most of the water and the solutes, anything below that renal threshold. Where does it take place? It takes place in the glomerulus and Bowman's capsule. So from the glomerulus into Bowman's capsule. How is the increased pressure maintained? It is maintained so that the pressure is generated from the heart. I mean, going into an organ, it's under higher pressure because it's coming from or a branch of the aorta. How it's maintained, however, is because of that difference in the afferent and efferent arteriole. The afferent arteriole has a wider lumen than the efferent arteriole, and that keeps that pressure high. What is not filtered out? Any of the larger molecules, so anything that is above that renal threshold, so proteins, for example, unless they're very small. What is the renal threshold? That is a molecular mass of 69,000. So anything below the molecular mass of 69,000 will be small enough to leave. What are podocytes? They are specialized cells, and their main function is to withstand the pressure so that the capillaries don't burst. Otherwise you'd be in a bit of a mess. Where are they found? They're found in that inner portion of the Bowman's capsule. What is selective reabsorption? It's absorbing specific solutes and water. Where does it occur? It occurs in the proximal convoluted tubule, and the processes involved, you've got active transport, facilitated diffusion, osmosis, co-transport, and diffusion. So pretty much all of the transport mechanisms, really. So if you want to have a think about where each of those things occurs, so it's active transport, facilitated diffusion, osmosis, co-transport, and diffusion. Have a think about where each of those occurs. So that's it for this one. Next time we are going to move on to the loop of Henle and the collecting duct, and you'll see there's a series of cartoons. There's kind of a little animation-y thing. You're going to build up like a cartoon strip for the loop of Henle.